Correspondence published. The 17th of March 2020. The Proximal Origin of SARS-CoV-2. Christian G. Anderson, Andrew Rambort, W. Ian Lipkin, Edward C. Holmes and Robert F. Gary. Nature Medicine, 2020. Cite this article. Note, the coronavirus is natural. It didn't came from laboratories. The coronavirus Note, the coronavirus is natural. It didn't come from laboratories. The coronavirus is natural. 3.42 meter accesses. 2 citations. 26,760 alt metric. Matrix details. To the editor, since the first reports of novel pneumonia, COVID-19, in Wuhan, Hubei province, China 1,2, there has been considerable discussion on the origin of the causative virus, SARS-CoV-23, also referred to as HCOV-19. 4. Infections with SARS-CoV-2 are now widespread, and as of the 11th of March 2020, 121,564 cases have been confirmed in more than 110 countries, with 4,373 deaths. 5. SARS-CoV-2 is the 7th coronavirus known to infect humans. SARS-CoV, MERS-CoV and SARS-CoV-2 can cause severe disease, whereas HKU1, NL63, OC43 and 229E are associated with mild symptoms. 6. Here we review what can be deduced about the origin of SARS-CoV-2 from comparative analysis of genomic data. We offer a perspective on the notable features of the SARS-CoV-2 genome and discuss scenarios by which they could have arisen. Our analysis clearly show that SARS-CoV-2 is not a laboratory construct or a purposefully manipulated virus. Notable features of the SARS-CoV-2 genome are comparison of alpha and beta coronaviruses identifies 2 notable genomic features of SARS-CoV-2, on the basis of structural studies 7, 8, 9 and biochemical experiments 1, 9, 10. SARS-CoV-2 appears to be optimized for binding to the human receptor ACE2, and II. The spike protein of SARS-CoV-2 has a functional polybasic furin cleavage site at the S1-S2 boundary through the insertion of 12 nucleotides 8, which additionally led to the predicted acquisition of 3 O-linked glycans around the site. 1. Mutations in the receptor binding domain of SARS-CoV-2. The receptor binding domain, RBD, in the spike protein is the most variable part of the coronavirus genome. 1,2. 6 RBD amino acids have been shown to be critical for binding to ACE2 receptors and for determining the host range of SARS-CoV-like viruses. 7. With coordinates based on SARS-CoV, they are Y442, L472, N479, D480, T487 and Y4911, which correspond to L455, F486, Q493, S494, N501 and Y505 in SARS-CoV-27. 5 of these 6 residues differ between SARS-CoV-2 and SARS-CoV, Figure 1a. On the basis of structural studies 7, 8, 9 and biochemical experiments 1, 9, 10, SARS-CoV-2 seems to have an RBD that binds with high affinity to ACE2 from humans, ferrets, cats and other species with high receptor homology. 7. Figure 1. Features of the spike protein in human SARS-CoV-2 and related coronaviruses. Figure 1a. Mutations in contact residues of the SARS-CoV-2 spike protein. The spike protein of SARS-CoV-2, red bar at top, was aligned against the most closely related SARS-CoV-like coronaviruses and SARS-CoV itself. Key residues in the spike protein that make contact to the ACE2 receptor are marked with blue boxes in both SARS-CoV-2 and related viruses, including SARS-CoV, urban E strain, B, acquisition of polybasic cleavage site and O-linked glycans. Both the polybasic cleavage site and the 3 adjacent predicted O-linked glycans are unique to SARS-CoV-2 and were not previously seen in lineage B beta coronaviruses. Sequences shown are from NCBI GenBank, accession codes MN908947, MN996532, A278741, Chi 417146 and MK 211376. The pangolin coronavirus sequences are a consensus generated from SRR 10,168,377 and SRR 10,168,378. NCBIBO Project PRJNA 573,298, 29,30. Full-size image. While the analysis above suggest that SARS-CoV-2 may bind human ACE2 with high affinity, computational analyses predict that the interaction is not ideal 7, and that the RBD sequence is different from those shown in SARS-CoV to be optimal for receptor binding 7,11. Thus, the high affinity binding of the SARS-CoV-2 spike protein to human ACE2 is most likely the result of natural selection on a human or human-like ACE2 that permits another optimal binding solution to arise. This is strong evidence that SARS-CoV-2 is not the product of purposeful manipulation. 2. Polybasic furin cleavage site and O-linked glycans. The second notable feature of SARS-CoV-2 is a polybasic cleavage site, RRAR, at the junction of S1 and S2, the two subunits of the spike 8, figure 1b. This allows effective cleavage by furin and other proteases, and has a role in determining viral infectivity and host range 12. In addition, a leading proline is also inserted at this site in SARS-CoV-2, thus, the inserted sequence is PRRA, figure 1b. The turn created by the proline is predicted to result in the addition of O-linked glycans to S673, T678 and S686, which flank the cleavage site and are unique to SARS-CoV-2, figure 1b. Polybasic cleavage sites have not been observed in related lineage B beta-coronavirus, although other human beta-coronavirus, including HKU1, lineage A, have those sites, and predicted O-linked glycans 13. Given the level of genetic variation in the spike, it is likely that SARS-CoV-2-like viruses with partial or full polybasic cleavage sites will be discovered in other species. The functional consequence of the polybasic cleavage site in SARS-CoV-2 is unknown, and it will be important to determine its impact on transmissibility and pathogenesis in animal models. Experiments with SARS-CoV have shown that insertion of a furin cleavage site at the S1-S2 junction enhances cell-cell fusion without affecting viral entry 14. In addition, efficient cleavage of the MERS-CoV spike enables MERS-like coronaviruses from bats to infect human cells 15. In avian influenza viruses, rapid replication and transmission in highly dense chicken populations selects for the acquisition of polybasic cleavage sites in the hemagglutinin HA, protein 6 gene, which serves a function similar to that of the coronavirus spike protein. Acquisition of polybasic cleavage sites in HA, by insertion or recombination, converts low pathogenicity avian influenza viruses into highly pathogenic forms 16. The acquisition of polybasic cleavage sites by HA has also been observed after repeated passage in cell culture or through animals 17. The function of the predicted O-linked glycans is unclear, but they could create a mucin-like domain that shields epitopes or key residues on the SARS-CoV-2 spike protein 18. Several viruses utilize mucin-like domains as glycan shields involved immunoevasion 18. Although prediction of O-linked glycosylation is robust, experimental studies are needed to determine if these sites are used in SARS-CoV-2. Theories of SARS-CoV-2 origins. It is improbable that SARS-CoV-2 emerged through laboratory manipulation of a related SARS-CoV-like coronavirus. As noted above, the RBD of SARS-CoV-2 is optimized for binding to human ACE2 with an efficient solution different from those previously predicted 7,11. Furthermore, if genetic manipulation had been performed, one of the several reverse genetic systems available for beta-coronaviruses would probably have been used 19. However, the genetic data irrefutably show that SARS-CoV-2 is not derived from any previously used virus backbone 20. Instead, we propose two scenarios that can plausibly explain the origin of SARS-CoV-2, I. Natural selection in an animal host before zoonotic transfer, and II. Natural selection in humans following zoonotic transfer. We also discuss whether selection during passage could have given rise to SARS-CoV-2. 1. Natural selection in an animal host before zoonotic transfer. As many early cases of COVID-19 were linked to the Huanan market in Wuhan. 1, 2, it is possible that an animal source was present at this location. Given the similarity of SARS-CoV-2 to bat SARS-CoV-like coronaviruses too, it is likely that bats serve as reservoir hosts for its progenitor. Although rat EG13, sampled from a Rhinalophysa finis bat 1, is 96% identical overall to SARS-CoV-2, its spike diverges in the RBD, which suggests that it may not bind efficiently to human ACE27. Figure 1a. Malayan pangolins, Manus javanica, illegally imported into Guangdong province, contain coronaviruses similar to SARS-CoV-221. Although the rat EG13 bat virus remains the closest to SARS-CoV-2 across the genome, some pangolin coronaviruses exhibit strong similarity to SARS-CoV-2 in the RBD, including all 6 key RBD residues. 21. Figure 1. This clearly shows that the SARS-CoV-2 spike protein optimized for binding to human-like ACE2 is the result of natural selection. Neither the bat beta coronaviruses nor the pangolin beta coronaviruses sampled thus far have polybasic cleavage sites. Although no animal coronavirus has been identified that is sufficiently similar to have served as the direct progenitor of SARS-CoV-2, the diversity of coronaviruses in bats and other species is massively undersampled. Mutations, insertions, and deletions can occur near the S1-S2 junction of coronaviruses 22, which shows that the polybasic cleavage site can arise by a natural evolutionary process. For a precursor virus to acquire both the polybasic cleavage site and mutations in the spike protein suitable for binding to human ACE2, an animal host would probably have to have a high population density to allow natural selection to proceed efficiently, and an ACE2 encoding gene that is similar to the human ortholog. 2. Natural selection in humans following zoonotic transfer. It is possible that a progenitor of SARS-CoV-2 jumped into humans, acquiring the genomic features described above through adaptation during undetected human-to-human transmission. Once acquired, these adaptations would enable the pandemic to take off and produce a sufficiently large cluster of cases to trigger the surveillance system that detected it. 1, 2. All SARS-CoV-2 genomes sequenced so far have the genomic features described above and are thus derived from a common ancestor that had them too. The presence in pangolins of an RBD very similar to that of SARS-CoV-2 means that we can infer this was also probably in the virus that jumped to humans. This leaves the insertion of polybasic cleavage site to occur during human-to-human transmission. Estimates of the timing of the most recent common ancestor of SARS-CoV-2 made with current sequence data point to emergence of the virus in late November 2019 to early December 201,923, compatible with the earliest retrospectively confirmed cases 24. Hence, this scenario presumes a period of unrecognized transmission in humans between the initial zoonotic event and the acquisition of the polybasic cleavage site. Sufficient opportunity could have arisen if there had been many prior zoonotic events that produced short chains of human-to-human transmission over an extended period. This is essentially the situation for MERS-CoV, for which all human cases are the result of repeated jumps of the virus from dromedary camels, producing single infections or short transmission chains that eventually resolve with no adaptation to sustained transmission. 25. Studies of banked human samples could provide information on whether such cryptic spread has occurred. Retrospective serological studies could also be informative, and a few such studies have been conducted showing low-level exposures to SARS-CoV-like coronaviruses in certain areas of China. 26. Critically, however, these studies could not have distinguished whether exposures were due to prior infections with SARS-CoV, SARS-CoV-2 or other SARS-CoV-like coronaviruses. Further serological studies should be conducted to determine the extent of prior human exposure to SARS-CoV-2. 3. Selection during passage. Basic research involving passage of bat SARS-CoV-like coronaviruses in cell culture and/or animal models has been ongoing for many years in biosafety level 2 laboratories across the world 27, and there are documented instances of laboratory escapes of SARS-CoV-28. We must therefore examine the possibility of an inadvertent laboratory release of SARS-CoV-2. In theory, it is possible that SARS-CoV-2 acquired RBD mutations, Figure 1a, during adaptation to pathogen cell culture, as has been observed in studies of SARS-CoV-11. The finding of SARS-CoV-like coronaviruses from pangolins with nearly identical RBDs, however, provides a much stronger and more parsimonious explanation of how SARS-CoV-2 acquired these via combination or mutation 19. The acquisition of both the polybasic cleavage site and predicted O-linked glycans also argues against culture-based scenarios. New polybasic cleavage sites have been observed only after prolonged passage of low pathogenicity avian influenza virus in vitro or in vivo 17. Furthermore, a hypothetical generation of SARS-CoV-2 by cell culture or animal passage would have required prior isolation of a progenitor virus with very high genetic similarity, which has not been described. Subsequent generation of a polybasic cleavage site would have then required repeated passage in cell culture or animals with ACE2 receptors similar to those of humans, but such work has also not previously been described. Finally, the generation of the predicted O-linked glycans is also unlikely to have occurred due to cell culture passage, as such features suggest the involvement of an immune system 18. Conclusions. In the midst of the global COVID-19 public health emergency, it is reasonable to wonder why the origins of the pandemic matter. Detailed understanding of how an animal virus jumped species boundaries to infect humans so productively will help in the prevention of future zoonotic events. For example, if SARS-CoV-2 pre-adapted in another animal species, then there is the risk of future re-emergence events. In contrast, if the adaptive process occurred in humans, then even if repeated zoonotic transfers occur, they are unlikely to take off without the same series of mutations. In addition, identifying the closest viral relatives of SARS-CoV-2 circulating in animals will greatly assist studies of viral function. Indeed, the availability of the RAT-EG13 bat sequence helped reveal key RBD mutations in the polybasic cleavage site. The genomic features described here may explain in part the infectiousness and transmissibility of SARS-CoV-2 in humans. Although the evidence shows that SARS-CoV-2 is not a purposefully manipulated virus, it is currently impossible to prove or disprove the other theories of its origin described here. However, since we observed all notable SARS-CoV-2 features, including the optimized RBD and polybasic cleavage site, in related coronaviruses in nature, we do not believe that any type of laboratory-based scenario is plausible. More scientific data could swing the balance of evidence to favor one hypothesis over another. Obtaining related viral sequences from animal sources would be the most definitive way of revealing viral origins. For example, a future observation of an intermediate or fully formed polybasic cleavage site in a SARS-CoV-2-like virus from animals would lend even further support to the natural selection hypothesis. It would also be helpful to obtain more genetic and functional data about SARS-CoV-2, including animal studies. The identification of a potential intermediate host of SARS-CoV-2, as well as sequencing of the virus from very early cases, would similarly be highly informative. Irrespective of the exact mechanisms by which SARS-CoV-2 originated via natural selection, the ongoing surveillance of pneumonia in humans and other animals is clearly of utmost importance. Note, the coronavirus is natural. It didn't came from laboratories. The coronavirus is natural. It didn't come from laboratories.